Okay, hello everyone. We are at the developmental biology department, but we are interested in the mechanics of development, which I guess kind of brings us on topic for this course. And so I'll tell you about the blastocyst, and the blastocyst is a structure which you've all gone through, and I'll tell you a bit more about it in a second. I know this doesn't work. Okay. So we're interested in development, and so many of you are maybe very familiar with the embryonic development. So embryonic development. So development is how you go from basically the zygote, which you can see on the left. Okay, so how you go from the zygote here, which is a cell with a relatively low complexity, right? And then you go into something which is way more complex, which has many, many cells. And those many, many cells, they are in a specific location. They have a specific shape, and they also have a specific function, right? The liver cells are within the liver, not within the heart. And so you have to organize all of the cells in 3D, and you have to give them the correct composition. And this is achieved by cycles of patterning and morphogenesis. So patterning is what gives the cells their identity, and this is often done through local cues, right? Positional information. So for example, here this cell knows that it's inside, and this cell here knows that it's outside, and those positional cue is what will determine the genetic program of the cells and will define their content, and will often define their behavior. The behavior may include changes in shape, and so that's morphogenesis. And so for example, here the embryo is becoming more compact, and so this is changing the shape, which often will give new interfaces. And those new interfaces will give new positional information, which will induce new patterning, which will then cause them to have a new behavior, and so on and so forth. And so through those cycles of patterning and morphogenesis, this is how you achieve the complexity that you can find in a newborn, for example. And if you've understood this, that's it. That's embryonic development. That's all what matters. Okay, and so in the lab, we're interested in the second part here, the morphogenesis. So how do you get the architecture of the body correct? This is still not working. Okay. Okay, there we go. And so when it comes to morphogenesis, so in the past 20 years or so, we have learned a great deal about animal morphogenesis. And so there was pioneering work in the late 90s and early 2000s in fly embryos. And so, for example, here you see this fly embryo, which is undergoing gastrulation with the mesoderm cells, which are initially in the same plane as the rest of the cells, the ectoderm, and they involute and separate into a distinct layer to form two different layers. And that's very important because our body has many different layers. And so this is mediated by changes in the contractility of the cytoskeleton of the cells, and this is coordinated by cell-cell adhesion molecules. And many of those aspects were discovered in the early fly embryo in the early 2000s. Fast forward another 5 to 10 years, and then many of those concepts have been validated in a vertebrate embryo, like a fish, for example. Here's a fish embryo which undergoes epiboly with this epithelium here, which is covering this big yolk cell here until it closes. Again, this is mediated by changes in the contractility of the actin cytoskeleton of those cells, and this is coordinated by cell-cell adhesion components of the cells. Fast forward another 5 to 10 years, and then you reach the mammals. And so here's a mouse embryo, for example, where the embryo is forming a lumen. So this is a fluid-filled compartment within the embryo. And again, the contractility of the cells and the adhesion of the cells is important to coordinate this morphogenesis. And so it took about 20 years to go from findings in fly, in vertebrate, all the way to mammals. And today I'll tell you about two different things. So one, I'll tell you about this lumen formation, and I'll also tell you about our own development. And so now that we know about mammals, what about us specifically? What about the human embryo? How do we shape ourselves? And so first part, I'll tell you about this, and then I'll tell you about the lumen in the mouse. Okay? So studying human embryos can be technically and ethically complicated. It's difficult to have access to material for experiments. But there's one time during our development which is accessible to experimentation, and that's pre-implantation development. So it is a phase of development that occurs before the embryo is implanting into the maternal uterus. And so during this time, you can culture the embryo in vitro entirely for the first 5 to 6 days. And this is what it looks like in a clinic, an IVF clinic, an in vitro fertilization clinic. So the embryo is now a zygote, and it will divide. These are cleavage cell divisions, so division without cell growth. The volume of the cell is just halved every time. And then it begins its morphogenesis. This took about 4 or 5 days. And here you have a structure, which is called a blastocyst. I let you read the statistics here. It's maybe not super important today, but just know that this is something which is very commonly known nowadays. It's 3 to 4% birth in France. So that's about 1 to 2% in the room today, for example. So it's becoming more and more common. So this is common, and so some of the embryos are never used for procreation, and some of those embryos are donated to research. And so you can actually access this material for your own experimentation within the legal limitations, of course. And so you can study this process now in the lab. And so these are just, again, the key steps that you've seen. And so the embryo goes from the zygote stage all the way to the blastocyst stage. And so the blastocyst has this very specific architecture with a tissue on the outside, which is called the trophectoderm. It's a squamous epithelium. It envelopes a fluid-filled lumen called the blastocell in green. And then there's this cluster of cells here in blue, which is called the inner cell mass. The cluster of cells here will give rise to all of the cells of the human body. Okay? These are pluripotent stem cells, which can differentiate into all of the cells that our body needs. The trophectoderm on the outside will only contribute to extramyotic tissues of the placenta. It will not contribute to the embryo at all. The job of the trophectoderm is to inflate this lumen and to attach to the uterus and implant the embryo into the maternal uterus. So without the formation of this trophectoderm, it's not implanting, right? So that's the end of it. Okay, and so we're interested in the morphogenesis of this embryo, and so I like to break down the shaping of the blastocyst in 3 different events. The first one is compaction, where the cells are initially loosely attached and then they form a tight structure. The second step is the formation of inner and outer cells. And this is obviously very important for the differentiation of the cells into either inner cell mass fate or trophectoderm fate. And then the last step is the formation of a fluid-filled lumen, the blastocell, which physically separate the trophectoderm, which is here, from the inner cell mass, which is there, and it breaks the radial symmetry of the embryo. And that's something which is also very important in developmental biology. We have specific axes in our body plan, and this is very important. And so this very first axis of symmetry, which is created here by the position of the inner cell mass, is more or less coordinating the dorsoventral axis of our body plan. Okay? And so this symmetry is broken very early on, and so very early on you have this axis which is transmitted then into the body plan of the future fetus. Are there any general questions about the basics of pre-mutation development? Yeah?
This breaking of symmetry is completely random at this stage, or is it
it's not,
by some...
No, it's not random. So I'll talk about this in the second part, okay. But it's not random. Yeah. Okay. Okay, so as I said, so we are interested in what shapes the human blastocyst, and so as I mentioned, the very first movement that we do is compaction. And so compaction is this process here. It takes about 20 to 30 hours. The cells just start to stick to one another. They form a compact structure. The surface of the embryo becomes smoother. The contacts are increased in size. This is a developmentally regulated adhesion process, which is triggered specifically at the 8 to 16 cell stage. If compaction is not going well, like for example for this guy, the embryo will have a lower implantation rate. This we know from clinical studies. We also know if this compaction, which is going well here, is delayed, there's also a lower implantation rate. So it's important to compact. What we also know is that sometimes all of the cells of the embryo are compacting together, and sometimes some cells are pushed out and not participating into the compacted part of the embryo. Those cells which are excluded will either be eliminated completely from the embryo, and that's it, they're just gone, or they are pushed to the surface and are part of this trophectoderm, which is only contributing to placental tissues. And this has been studied in vitro, in clinical studies. We know that some of those cells which are excluded tend to be aneuploid. They tend to have extra or missing chromosomes. And we also know from natural pregnancy, not in vitro grown embryos, that in human, during human pregnancy, the placenta is a tissue which is full of aneuploid cells. It's just something which happens naturally. The placenta is a very unstable tissue, and there's just plenty of aneuploid cells. And there's this idea now that basically the job of compaction is to push out to the extra embryonic tissues any cells which would be unfit, having too many chromosomes, too few chromosomes, and so on and so forth. It would be a filter which would just get rid of cells which you don't want in your body, but you can afford to have in the extra embryonic tissues. But we don't know how those cells are excluded. What is the mechanism that drives the exclusion of those cells? Okay, so if you consider now the different mechanical properties of the interfaces that we have in the embryo, so you can sum it up to two cells contacting each other. They will have distinct surface tensions at their distinct interfaces. And so here you have a cell-cell contact with a tension gamma cc and a tension gamma cm at the cell medium interface. And if you increase the tension gamma cm, you pull at the edges of the contact, you enlarge cell-cell contact. If you relax the cell-cell contact, you reduce the gamma cc, the angle of contact will also increase. And basically, you can either drive compaction by relaxing cell-cell contact or by pulling harder on the edges of the contact. Okay, it's very analogous to a wetting transition, and this is how we approach this problem here. And so all what we need to do now is measure those surface tension and the angles of contact. And the way we measure the, I have to really stop clicking on this. The way we measure the surface tension is by micropipette aspiration. Okay, I don't know how many of you are familiar with this, but basically we use a pipette to apply pressure at the surface of the cell. And when the deformation here is corresponding to a hemisphere, we can apply the Laplace law, and it relates the surface tension gamma cm to the pressure of the pipette and the shape of the pipette and the shape of the cell. Okay? Once we have the tension of two contacting cells, we can also measure the angles of contact and apply another force balance equation, which gives us the tension at the cell-cell contact as a function of the tension gamma cm's and the shape of the contact angle. And so the assumptions on this that the cells are behaving like soap bubbles, like a viscous material, a viscoelastic material, and that the tensions are homogeneous at the cell interface and at the cell-cell contact. We are not considering any heterogeneities in the tension at the interfaces. This assumption is to some extent true, and probably in some cases not true. Okay, so this is how it looks like in the mouse. So we had extensively done those measurements in the mouse before we could get our pipettes into human embryos, and this is how it looks like. You basically aspirate each cell successively, and you can continue doing this throughout the entire process of compaction because we are not damaging the cell by deforming them. And so you can map the tensions of all of the interfaces in space and time. Okay? Now, this is a human embryo which is donated to research, brought to the lab. This embryo here comes from infertile couples. We do not know whether it's a problem with the mother, with the father, or with the embryo per se. We do not know if this could ever become a healthy individual. We will never check, right? But that's one issue in the experiment here. We do not know if this is a healthy material, okay? We are also taking those embryos about 15 to 20 years after they've been conceived. They've been frozen for this entire time. We don't really know if the freezing was an issue or not. Then we put them into a dish. We shine light on them. That is not good as well. We also remove the eggshell. There's an eggshell normally around it. And so we dissect out the eggshell. That's also not so nice. And then we apply micropipette aspiration on those embryos. Okay? So we consider the embryos which make it. And by making it, I mean that those embryos that we consider are those which divide normally, compact normally, and then form a blastocyst. And there we stop. And so here you see developing under the microscope, it forms a lumen. And here you go, you have a blastocyst with an inner cell mass, if you can see it. And so this is one of the criteria that we use, right? We look at them, we consider them in the analysis when they have made a blastocyst. Doesn't mean that they are healthy, but that's our standard. Okay? Okay, and so now what happens to the embryos is that during compaction, the angle of contact is increasing. It increases about the same way in the mouse and human, and about the same way in the embryos grown in the lab and those which are observed in the clinic. During this time, the tension gamma CM is growing from 500 to 2,000 piconewton per micrometer, so it's fourfold increase. At the same time, the tension at the cell-cell contact is remaining steady. So what seems to be driving compaction is this increase in tension at the surface of the embryo, not a relaxation of cell-cell contact as you would expect, for example, from two soap bubbles which would adhere to one another. Okay? So for this, we had synchronized the embryos to the time of third cleavage, so when the embryo is going from 4 to 8 cell stage. Sometimes, because those embryos were frozen after the 8 cell stage, we don't have this time, and so we cannot synchronize the embryos. And so here are 8 embryos, but if we now just plot the tension gamma CM as a function of the contact angle, regardless of the stage at which we took them, we have now 13 embryos, right, which is a bit more. And then we have a good correlation between the tension gamma CM and the angle of contact, which suggests that you can nicely predict the tension. You can actually predict the shape by the tension at the cell unit phase. And this is not the case for the tension at cell-cell contact, which remains, again, constant throughout all the different states of compaction. So what seems to be driving compaction is this increase in tension gamma CM at the cell unit phase. As I mentioned, we also had measured the mouse embryos, and we can now compare those two species. So the mouse here compacts in less time, okay, than the human, and something that we knew before. We knew before that the compaction is faster in mouse than in human. The increase in tension gamma CM is conserved, right, between those two species. The magnitude is different, so here it doubles, and for human it is multiplied by 4. The tension at the cell-cell contact of mouse embryos is decreasing a little bit by about 1/3, and this is contributing to compaction. And so now we have the same movement. If we measure the angles of contact between those two embryos, they're very, very similar. The movement is the same, but the underlying mechanical strategy is different. Okay? We rely entirely on this increase in gamma CM, while the mouse is compacting thanks to 3/4 to the increase in gamma CM and 1/4 to the relaxation of cell-cell contact. So it's something which is, then the strategy is qualitatively conserved with this increase in gamma CM, but quantitatively there are some differences. We can also compare them on a phase diagram where we have here the tension at the cell midpoint phase, and then on the y-axis the tension at the cell-cell contact. And color-coded is the ratio gamma C over gamma CM, which corresponds to the cosine of the contact angle. So to compact, the embryo has to go from the green area to the orange area. And you see that based on so based on the angle that the embryo is taking, so the human embryo is going all the way to the right, increasing gamma CM, that's how it compacts, and the mouse embryo is decreasing a bit gamma CC. And now if you start to consider like what would be the fastest way to compact, you can think maybe this is the shortest path. So a mix of decreasing gamma CC and increasing gamma CM. And neither the mouse or the human would be following this potential shortest path, but clearly the human embryo is not as close as the mouse one. So it looks like we are less efficient at compacting than the mouse embryo. Why that is, whether that's important, I don't know. But that's just something we see. We also could get our pipettes on rabbit and cow embryos. These are very preliminary measurements. But what we see as well is that so there may be some differences again in the mechanical strategies which underlie the compaction of those different species. What I just would like to point out is the scale of the tensions here. So mouse, rabbit, and cow have tensions in the ranges of hundreds of piconewton per micrometer, and human embryos have tensions in the ranges of thousands of piconewton per micrometer. So somehow we have this very high tension early on. Whether this is important and meaningful, I do not know. It's just something that we see. Yeah.
As you explained earlier, the human embryos that you use are
frozen for a very long time.
Fresh.
Uh-huh.
So, and I guess in the case of cow and rabbit, you don't have this problem. So
yes.
I wonder if those
yes.
quite extreme mechanical differences that I don't know if I would expect them within mammals. You know, are they? I know you can't answer that, but do you think it's related to that?
Yes, so there could be many different reasons. So one is differences between species. Then it could be differences in how long they were frozen. Then the way they were conceived. These guys were conceived naturally. These guys as well. The cow was conceived by in vitro fertilization. So we recover the ovaries from the slaughterhouse, and we fertilize them in vitro. This guy, there's no slaughtering, and it's conceived in vitro as well. So this could be important. The medium is not exactly the same either. The temperature at which they are grown is not the same. The atmosphere at which they are grown is not the same. So cow and human are grown at 5% oxygen. That's required for the development. This is not the case for the other two. So there could be many different reasons. So I'm not claiming in any way that this is not due to a technical difference. But I'm just trying to make a point that there's probably something to look into there about the differences in the mechanical properties of different species in general, where we could maybe learn something about the different strategies that are adopted by different species, and using mechanics as... So the good thing with mechanical properties is that they have a unit. You can compare species directly, which is not something which you can do easily with many other metrics. Like if I ask you to, how many actin monomers there are in this cell, and this cell, and this cell, no one can give this answer.
Yeah, but
And so we
agree
can...
that in order to have a meaningful comparison,
Yeah, we will need to...
we would have to standardize
the way they are conceived. Exactly. So there are things we can do. So, making those guys in vitro, this we can do. Culturing the mouse embryo in the same medium as the human, we can do. Culturing them at 5% oxygen, we can do. What we cannot do is freeze them for 15 years.
Yeah, sure. Thanks.
Any other question? Okay. So,
oh, sorry.
yeah, sorry.
I have a question actually, but it was more on the start of the
Uh-huh.
talk. So if I saw correctly, the compaction happens at like 16 cell stage, more or less. And usually for monozygotic twins, it happens at 8 cell stage.
How do you know this?
I saw this in an article, but
Okay.
usually.
Okay.
So I was wondering if there is a link between compaction and the possibility of dividing.
So to my knowledge, we do not know what causes twinning. So twinning is something which is very common in the human species as compared to other mammalian species, and yet we still do not know, to my knowledge, how this is done. And whether this could be a mechanical problem, I guess so. Whether it's compaction, I don't know. Okay. Yes. Okay. So now we can go into what drives these changes in surface tensions. And so initially, compaction was described as an adhesion process. The adhesion molecule, E-kedrin, is what mediates compaction. E-kedrin was actually discovered by studying compaction. Here is an embryo which is cultured with an antibody which blocks E-kedrin. That's the very experiment where E-kedrin was discovered. And the idea is that the E-kedrin adhesion molecule, which is a transmembrane adhesion molecule, binds to the actin cytoskeleton, stabilizes the adhesion complex, and then by some thermal fluctuation, at the edge of the contact, you would just stabilize the contact by zipping it up like this. That would be the idea that would decrease gamma CC by increasing adhesion between cells. But we also know that animal cells can change their shape thanks to tensions generated by the actomyosin cytoskeleton. You probably heard about the actomyosin cortex? Okay. No? No. Okay. So it's a thin meshwork underneath the plasma membrane of cross-linked actin filaments. The myosin motors are sliding the filaments against each other until they are under tension. And then this tension is transmitted to the cell surface by linking this meshwork to the plasma membrane. And it's a very important determinant of animal cell shape. A good way to visualize this tension is to cut the network. So here, this is the actin cytoskeleton that you can see here. And in this experiment, they cut the cortex with a laser, and you can see that it snaps. And so this snapping is just, I think, a nice visual way to see the tension within this network, which can then change the shape of cells. Okay, and so we know that it's what drives cell shapes in fly, in mouse, in fish. There's no reason to suspect that it's not acting in human. So, behold, you block actomyosin contractility, and the embryo is less compacted. It's a mild decompaction in this case. The tension gamma CM is dropping by a factor 3 if you treat the embryos with an inhibitor of myosin activity. And the tension at the cell-cell contact is also dropping. So there is quite some contractility at the cell-cell contact. Okay? And because the drug is inhibiting contractility everywhere, at the same phase and the cell-cell contact, by reducing gamma CC, we are actually promoting compaction and by decreasing gamma CM, we are inhibiting compaction. So there's a bit of a mixed effect, and the decompaction is mild by about 30 degrees on average. Okay? But what we conclude from this is that atomizing contractility is what generates this tension gamma CM and gamma CC. Okay? So because gamma CM is what drives compaction, atomizing contractility is what drives compaction in human embryos. We also looked into adhesion, because adhesion is supposed to be the main thing. And so we block cadherin-mediated base adhesion using a medium which does not have calcium. So the K in cadherin is for calcium-dependent adhesion molecule. And if you chelate all the calcium from the medium, the embryo decompacts. So that's something which is very well known. But when we look at the tension, gamma CM, it's not changed. The tension gamma CM, which is what drives compaction, is not depending on the activity of cadherin adhesion molecules. The tension gamma CC is increasing. You remove adhesion molecules, the tension is increasing at the cell-cell contact. That's also something which you could expect. But so there is no change in gamma CC during compaction, normally. So this is not what's happening. What we conclude from this is that what drives compaction in the control situation, in black, is this increase in atomizing contractility, specifically at the cell-cell contact. Then you take those embryos, you treat them with an inhibitor of myosin activity, and they move back to this region of low compaction. And you block adhesion. They also can be blocked from compacting. But what you can see is that those EDTA and ML7 conditions, they are in very different areas of the phase diagram. Low contractility corresponds to regions of low tension, both gamma CC and gamma CM. Low adhesion corresponds to regions of high tension, high gamma CM, high gamma CC. So same aspect, decompacted, but different mechanical signature. Okay? And so now what we're going to do is we're going to use this mechanical signature to try and find out why some embryos fail at compacting. Okay?
So there
Yeah.
are two questions here. So
Yeah.
you want to interrupt
Yeah.
before, but so our questions from Camilo Andino, which is a remote attendee. So thanks for the nice talk. So the first question was asked a few minutes ago. So I was wondering, is it known in these different species what is the mechanism responsible for the change in the surface tension during compaction?
No, nothing is known about the compaction of those species.
And as a follow-up question, is it known what is the ratio between inner and outer cell mass in the different species after compaction?
So for the mouse, the compaction happens before cells come in and out. For human, it's more or less at the same time, and there is maybe one cell in for human embryos on average, I mean, as far as we could see. For the cow and the rabbit, I don't think this has been studied.
Thank you.
Okay? Okay. I'll speed up a little bit so that I can talk about the rest. So basically, again, we're interested in why sometimes it fails, right? And so this is a normally compacting embryo, and here it's an embryo which did not compact. We want to make sure that we are not working with dying embryos, and they could be doing nothing because they're just dead. And so we just make sure that those cells are still dividing. Okay? This is our criterion to say that the embryo is still alive. And we have taken a cutoff at 140 degrees. So if there's not a single contact angle which reaches 140 degrees in this embryo, we consider that it failed compaction. Now, if you compare the tension of those two types of embryos, those that do manage to raise the contact angle above 140 degrees, so which do increase gamma CM and keep gamma CC constant, what we see that those that fail, they do not increase the tension gamma CM. So the scenario which is shown there is basically corresponding to a defect, not in adhesion, but to a defect in contractility. Okay, that is the mechanical signature of an embryo which failed to increase contractility. And so that is a more likely explanation than a defect in adhesion like what is suspected in the literature. So that's when adhesion can fail everywhere for all the cells. But as I mentioned to you, there are also those cells. Okay, well, this is just a different way of showing the data. Because as I said, here we need to synchronize the cells to a certain cleavage, and we only had 4 embryos, right? That's very few. If you pull all of them together, then you reach 9 non-compacting embryos, which is maybe a bit more reassuring. But that's the same thing, okay? And so then we have those cells which are excluded, which potentially are saving the embryo from having some aneuploid cells. And so again, we take an embryo with unequal compaction as an embryo where some of the contact angles are above 140, and then we consider that the cell is excluded is by the end of compaction, this cell has a contact angle lower than 120. Okay, and so this is what it looks like. You have here an embryo before compaction. It's very hard to tell which cell is going to make it and which cell is not going to make it. They all look very similar. They all look not very good, actually. Then you have this compacted mass in the center, and you have those cells which remain at the surface. So here, this one has 4, at least, excluded cells. It's quite many. I show it as an example. Typically, you're more going to have maybe more like 1 or 2 excluded cells at most. Okay? This one was not very happy. And so here are the cells which are compacting. They increase their contact angle in purple. They increase the tension gamma CM in red. They keep gamma CC constant. The cells which are not compacting, they keep gamma CM low. So they definitely also show sign of a defective contractility. On top of it, they may have also an increase in gamma CC, which would show, in addition, a defect in adhesion. And so we are still looking into this. But they definitely show signs of defective contractility. If you now start to consider the different scenarios of how do you exclude a cell with defective gamma CM or defective gamma CC. So the first video shows you an embryo. So that's a collaboration with Hervé Turlier at the Collège de France. This is an embryo which does compact, and all cells are increasing gamma CM and keeping gamma CC low. This is an embryo where the pink cells keep gamma CC low, but also gamma CM. And what you can see is that because the surrounding cells, they have a very high gamma CM, they actually stretch the neighbors over themselves. So having a low gamma CM in this cell here does not give us the same shape as we observe in the experiment. If we want to have an excluded cell, we can have a low gamma CM in the excluded cell, but it's important to have a high gamma CC at the interface between the healthy part of the embryo and the unhealthy part of the embryo. And so one way to have this high gamma CC at the interface between excluded cells and non-excluded cells would be to have a high contractility. And the high contractility is unlikely to come from the cell which is becoming excluded, because gamma CM suggests that they have a low contractility. So what this suggests is basically that the exclusion is not just something which is happening because of the excluded cell, but something which is coming from a collective behavior, where the healthy part of the embryo is contracting the contact at the interface with the cells which are in some way recognized as not healthy. And so this is what would come from the prediction of this theory, and to some extent from the measurements. But because those cells are quite rare, we will, I think, never manage to get enough statistics to clearly show that gamma CC is increasing. But we propose that this is something which comes from an internal defect of the excluded cell, and also a contribution of the surrounding cells to exclude the cells which is potentially going to harm the embryo. I'm done with compaction. Do you have questions about all this? Yeah?
Related to the exclusion, has it been generally checked, like if aneuploidy cells have a different level of E-cadherin expression or something like this in terms of their adhesion?
So the question is about the molecular composition of the excluded cells. Yeah. There is no such study. All what has been studied is how many excluded cells do you have, when do they get excluded in development, and they have checked the ploidy, and that's it. Okay, yeah?
When you look at the curves and the error bars, or the embryos that do not compact, it seems to have some kind of oscillations if the error bars are as small as...
Yeah. Yeah, so as I mentioned here, so we have a bit of a sampling issue. So it's, so this embryo is a bit extreme. There are 4 excluded cells. Typically, it's more like 1. And as you can see, we have 6 embryos in which we could detect excluded cells. So these are coming from 23 measurements across 30 hours. And we have binned the embryos by bins of 5 hours. So this dense, they're probably just coming from this low sampling. I would not trust those so much. I think,
yeah. Because the error bars are kind of small.
Yeah, it's the SEM. It's the SEM, yeah. Yeah. So, I mean, to some extent, I'm very happy that there is not too much variability between all those different human embryos which come from different patients and so on. We could have expected a lot more. So I'm happy that it's kind of tight for those guys, for which we have quite a few measurements. Nevertheless, it's 6 embryos, which, how much can you conclude from this? I don't know. So I think the trend over time is something we can trust, but the small, bits, I'm not sure. Okay. I'm happy to get more embryos. It's just sometimes complicated. Okay? Okay, so let's switch to something else. Just take home message. Whoop. Compaction is mediated by this increase in tension, which is conserved in evolution, and is mediated by actomizing contractility. If it's defective, compaction will not happen, and you could potentially then also have cells which are excluded because of this low contractility. That's kind of what we could learn, I think, from the study with human embryos. And this is, to a large extent, something which we have started because we kind of knew that it would happen this way. We have extensively characterized the mouse embryo. There's, to some extent, little surprise, but it's just nice to see that we can confirm some of those aspects in human. Now let's go into a system where we can have many embryos and get some robust numbers. So I'll tell you about something different, which is the formation of the first mammalian lumen, which breaks the symmetry of the embryo. So this lumen forms in this way. So the mouse embryo now, so it's all mouse now. The mouse embryo here has about 32 cells. Okay, so it has gone through its 5th cleavage, and the lumen is going to appear on this corner here and break the contact between those trophectoderm cells and those inner cell cells. You want to break contact, but you don't want to break any contact. You don't want to break contact between trophectoderm cells, otherwise the fluid will leak out. And you don't want to break contact between the cells of the inner cell mass, otherwise it will scatter everywhere inside the blastocyst, and this could lead to twinning, which you may want or not, but you may want to control it, right? So you want to make sure that what breaks are the heterotypic contact between trophectoderm and ICM cells. Okay. So the basics of lumen formation in animal cells is the following. So you have a polarized epithelium with an apical side, a basolateral side in purple and blue, and what has been studied are mostly those cells here which form apical lumen. So the lumen forms on this red area. This red area is characterized by very low, if any, adhesion. The adhesion molecules are here with the cadherins at the cell-cell contact, and the adhesion to the matrix on the basal side, so all the integrins and so on and so forth, on the basal side, away from the lumen, away from the interface that you want to separate. Sometimes this apical surface here even contains proteins which are heavily negatively charged, like podocalixin, which is a mucin, and this will actually repel the apical membranes from one another. Okay, so it's very unadhesive. Then how do you bring the fluid? It's with polarized transport. So the cells are transporting osmolyte from one compartment to the other, and then the water will follow the osmotic gradient and go from the outside of the cyst into the lumen. And then you want to make sure that everything is tight, and this is provided by tight junctions, which are in green, which prevent the osmolyte to go away and the water to go away and contains the pressure as well. Okay? This is the same machinery which is at play in the mouse embryo, but the polarity is inversed. So in the mouse embryo, the apical side is on the surface, and the lumen, which forms in the very same way, with the same machinery, forms on the opposite side, on the adhesive side of the tissue. So the side which contains the E-cadherin adhesion molecule. And now we don't know how the lumen can form if you have to break adhesive contacts. Okay, and so what we want to understand is how do you form a basolateral lumen, because this was not known. And as I mentioned briefly, we are also interested in how you position this lumen, because the position of the lumen will define the first axis of symmetry of the embryo, which defines this embryonic pole, which will define the site of implantation, and then the dorsal-ventral axis of the embryo. Okay, so if you film this event with higher spatial and temporal resolution, this is what you see. This is a 32-cell stage embryo. There are inner cells and outer cells, and the fluid starts to accumulate at cell-cell contacts. All cell-cell contacts are transiently broken with small water pockets which accumulate at the cell-cell contact, and then the water pockets are, in a more or less synchronous way, disappearing, and only one very large water pocket remains and becomes the blastocell. Okay? So we have two types of what we call now microlumens, so tiny lumens, right? We have those which are exclusively between two cells. We call them bicellular microlumens. So this is really just a little capsule of fluid which is trapped between two cells. And then we have the multicellular microlumen, which are the contact edges. So if you do a confocal slice like here, you see it between three cells, but it's a 3D sphere. And so you have to imagine that this multicellular lumen network is running throughout the embryo, okay, between many more cells than three. The bicellular microlumen and the multicellular microlumen, they behave very similarly in the sense that they first grow and then they start to shrink. The small ones, the bicellular one, they grow and shrink first, and then the multicellular microlumen, which are bigger, do the same thing. And you can see that here. They're basically doing the same thing, just at different scales. So these guys, the bicellular microlumens are just smaller and slower, and they happen a bit earlier. Okay? So we thought that those structures resembled something which had been observed in this experiment here, where what they have done is they have taken a monolayer, so now it's a 2D surface, and they have plated it on a circular pattern. And this is now plated on a hydrogel, and they stretch the hydrogel because they wanted to study the effect of stretching in epithelium. And then they release the stretch, and when they release the stretch, they see those fractures. And this is because the hydrogel is stretched, it loads with medium, and then when they release the stretch, the medium is squeezed out of this sponge-like material and is flushed through the epithelium. And when the fluid is flushed through the epithelium, it breaks the cell-cell contact. So this is hydraulic fracking, the same thing that you use to extract gas from the earth, right? And so we thought that those structures somehow resemble what we observed. So here it's a 2D structure, and here it's a 3D structure. And they had estimated the amount of pressure that you need in the fluid to break open those cell-cell contact. And it's in the range of a few hundreds of pascals, okay? So we first measured the pressure inside those lumen, and so this is a micropipette aspiration that we use to also measure the pressure inside the lumen. We measure something in the range of 200 Pascal. It also depends on the activity of actomycin. So the fact that the epithelium is squishing the fluid, pressing onto the fluid, could also build up the pressure. And so we have pressures which are in the correct range for hydraulic fracturing to happen. And with this idea of hydraulic fracturing, we expect that the adhesion molecule are going to be displaced. Okay? And so we image the adhesion molecule, and here is E-cadherin GFP in green, and the membrane here is in magenta. And what you can see at a global scale is that when the microlumens are appearing in the bottom left corner, the adhesion molecule go from an homogeneous signal to something which is patchier. If you zoom in, you can see this. E-cadherin is in green. It is along the cell-cell contact, and when the microlumens are forming, the E-cadherin is relocalized to the edges of the microlumen, so it's pushed out, right, to the side. Now the question is this reorganization of E-cadherin something which is happening because of the fluid accumulation, or would it have happened anyway? Is the fluid accumulating because E-cadherin is leaving a weak spot, or is it the fluid which is pushing away the E-cadherin? And so we block the fluid accumulation, and we see whether E-cadherin is relocalized or not. And so we have three different ways of blocking the fluid accumulation, and what we see is that if you block fluid accumulation in those three different ways, E-cadherin stays more homogeneous. Okay? So you need fluid accumulation to relocalize E-cadherin to this patchy foci. So this is compatible with the idea that the adhesion is broken by the fluid. What we would also expect is that if you were to weaken adhesion, then the fluid would have an easier way of breaking in and opening the lumen. And so we used a mutant for E-cadherin, which lacks the maternal allele, but it still has a paternal allele. And that's enough adhesion to make a blastocyst, but it has less E-cadherin in total. Now what we see in those mutants is that they form the blastocell faster, so the fluid accumulates faster. They still form microlumens, and those microlumens are increasing in size faster than in a wild-type embryo. And then the second phase, the coarsening, is also a bit faster, but not so much. Okay? So the idea is that now it's easier to break open the contact, and so the fluid rushes in faster. Again, which is compatible with this idea of hydraulic fracturing. And so now we have a way also to pattern adhesion within one embryo. So if we take wild-type cells and mutant cells and we bring them together, we can make an embryo where half of the embryo has a normal amount of adhesion, and then the other half has a weaker adhesion. And so then we expect that the fluid will follow the path of least resistance and will go into the half which has less adhesion. And so here, this is a chimeric embryo with wild-type and wild-type, okay? And what we see is that the lumen forms somewhere, and here this is quantified. So half and half are going into the ICM. So the inner cell mass is composed of both wild-type cells. Then we have two types of trophectoderm, okay? There's a trophectoderm which is lining the lumen, which is called the mural trophectoderm. And then we have the trophectoderm which is lining the ICM. And this one is called polar trophectoderm. And we see no bias between the two different wild-type cells. If you now take the mutant cells, they both contribute to the inner cell mass, but you can see here in this example that the lumen is completely surrounded by mutant cells. Okay? And so the mural trophectoderm is primarily composed of mutant cells, and the mutant cells are depleted from the polar trophectoderm, which is away from the lumen. Okay? So by patterning cell-cell adhesion, you can position the lumen, and this is sufficient then to position the first axis of symmetry of the embryo. And this will determine then its implantation site and its dorsoventral axis. Okay? So this is a mechanical patterning. Okay? The way this symmetry is broken is mechanical. Okay, so the idea is now that there's hydraulic fracturing and that this is breaking cell adhesion and that this is positioning the lumen. And so we looked for signs of adhesion pattern in vivo, in a regular embryo, not in a manipulated embryo. And we looked at, for example, where the microlumens are appearing. And so we expect that, for example, here it broke a bit faster. And so probably the lumen is going to be there. But of course, it's not. It goes on the other side. And we looked at any kind of cue that we could see. Like we looked at the distribution of adhesion molecule. Can we see a pattern in relationship with the final position of the lumen? Nothing obvious, nothing clear. We could not find any bias in how fast it opens and how big it opens or how much adhesion you have. So you can manipulate adhesion and it will influence the positioning, but we could not see any evidence of a patterning. So we looked at the second phase, which is the coarsening, so how the microlumens are disappearing. And so we looked at it first at higher temporal resolution. And here you have two multicellular microlumen, which are contained between multiple cells. And then you have those here, which you may be able to see when the movie is going to play, which are trapped between two cells. And we are imaging now every 5 seconds. So it's a much faster imaging process. And you can see a bit of a pulsation going on. And you can see that these two multicellular microlumen are growing over the course of the movie. And the ones which are at the cell-cell contact, they slowly disappear. There is no coalescence, except for those two which are directly next to each other, which eventually fuse. It's not like those guys are coming in contact with the big one and fusing with it. Instead, they just seem to lose volume and disappear. And so this is a process which is akin to Ostwald ripening, where you can have two liquids which are mixed in an emulsion, and then because of surface tension, the small droplets will empty themselves into the big one because of their smaller radius of curvature, because the Laplace law connects the surface tension and the pressure and the radius of curvature. And so we are taking the same this as an analogy for what may happen in the embryo, where we have the microlumens which are connected by the intercellular space. And because these guys would have a higher pressure due to their smaller size, they would empty themselves into the bigger microlumen. And so you can build simulation of this. This is again Hervé Turlier. And you take this smaller microlumen, the tension is the same for both. Because it's smaller, it will empty itself into the bigger one because it has a higher pressure. But cells are not like a vinaigrette, and they can actually tune the surface tension at specific interfaces, as we have seen. And you could imagine a situation where this one here is smaller than this one, but this one now has a higher tension because it has more actomycin, for example. And now you can actually reverse the fluid flow. Okay, yes. And now the big one is emptying itself into the small one. Okay? So in addition to the surface tension and the size, which can be modulated to trigger this pressure gradient, the microlumen is forming a network, and the connectivity of this network is going to be important. Because the microlumens which are central to the network may be better connected, and then may have more reservoir to exchange fluid with, while those at the periphery will have different dynamics in their fluid exchange because of their fewer neighbors. Okay? And so if you now build an artificial network where you have microlumen trapped between ICM cells and those which are trapped between ICM and T cell, and you let that coarsen, the ones which are better connected on the inside, if the tension are equal, are going to win, and the lumen will form within the ICM, which never happens in the embryo. This is not what happens. Okay? But if you make the ICM have a higher tension than the trophectoderm, which are the surface, then the lumen will form at the trophectoderm ICM interface, which is what we observed. We knew beforehand that the ICM cells are more tense than the trophectoderm cells. So it was not a surprise. But we looked for more evidences that the ICM cells have a higher tension at the trophectoderm, and so you can see that by looking at the curvature of the interfaces, for example. This is a control embryo, and you can see that between two ICM cells, the interface is rather flat. Between two trophectoderm cells, the interface is rather flat, but then when you look at the trophectoderm ICM interface, the ICM cell is bulging into the trophectoderm. And this curvature is gone if you block actomycin contractility with this inhibitor here, which is called blebistatin. And so the interfaces become flatter between trophectoderm and ICM if you block contractility. And so because these guys here in the ICM are more contractile than the cells of the trophectoderm, they bulge into them. Those curvatures, you can also see them at the level of the microlumens themselves. So microlumens can appear at the trophectoderm interface or at the trophectoderm ICM interface. They can be more or less symmetric, the microlumens. They tend to be more symmetric between trophectoderm cells than between trophectoderm and ICM cells. So the one which are at those heterotypic interfaces, they are more often asymmetric. And when they are asymmetric, they bulge into the trophectoderm cell rather than the ICM cell, which again indicates either difference in tension or difference in pressure or both. Okay, so this pattern of contractility and this pattern of mechanical property is compatible with the idea that the coarsening would lead to the positioning of the lumen. And so now what we need to do is again manipulate the mechanical property and see if if we can manipulate the positioning of the lumen. And so we again built chimeric embryos to create a pattern. Now we did not want to have a pattern of adhesion, but a pattern of contractility. And so we used those myosin mutants, which are heterozygous. So they still have the paternal allele of this gene here, MYH9. They have a contractility which makes them have a surface tension about threefold lower the one of a wild type, as we have measured. And so you can create this embryo with a pattern tension with one side which has a lower surface tension than the other. And so what we expect is that when the adhesion is broken and the fluid starts to accumulate throughout the embryo, we expect that essentially those cells here are going to squish the fluid away from their interfaces more efficiently into the region which is more deformable. And we expect that then the fluid will go there. And so this is, of course, what happens. So this is a wild-type chimera. You have the same kind of quantification as you have seen before. And now this is a wild-type myosin mutant chimera in green, the myosin mutant cells. The myosin mutant cells, they have issues dividing, right? Because you need myosin to divide. So they have fewer cells, but those fewer cells are more often located at the mural trifectoderm, which is lining the lumen. And so this is a quantification here. They are also depleted from the inner cell mass. This is also something that we knew. And so in the same way that patterning adhesion can lead to the positioning of the lumen, patterning contractility is also sufficient to position the first axis of symmetry of the mouse embryo. And because we know that both of them, both adhesion and contractility, can position the lumen, but we did not find any evidence for a pattern of adhesion, but we did find a pattern of contractility. We think that this is what drives the positioning of the first lumen, and then the positioning of the first axis of symmetry. We think it's mediated by contractility. And so at the moment now we are looking into the fine cellular events which would lead to the positioning of the lumen. And we have identified some processes which are called BLEBs, which are mediated by ectomyosin contractility, which could be involved in the positioning of the lumen. But I'm not talking about this today. Okay, so I talked about two different stories. So the first one was about human embryo development. And so this is carried out in the lab by Julie Firmin. She's a medical doctor, and she's doing a PhD in the lab in co-supervision with Catherine Patra, who is the head of the fertility clinic at Institut Cochin. This is also in collaboration with Hervé Turlier, with a theoretical physicist at the Collège de France. He also did some of the simulations together with the student, Mathieu, for the positioning of the lumen. Many of the experiments of the lumen positioning were done by Francesca and by a postdoc, Julien, who is not on this picture anymore. And the rabbit and cows, this is coming from INRA, from the lab of Véronique Duranton. Thank you. From the chat.
I was really intrigued by this fact that this compaction at the beginning could sort of exclude the aneuploid cells. And then the fact that contractility of this actomyosin network or mesh could play a role. How would you connect these two things? I mean, I could imagine that aneuploidy will be connected through spindles, so kind of the microtubule part of the skeleton. So how would you connect that to actomyosin?
Yeah, so we still have to check whether those cells which are excluded are indeed the aneuploidy ones. So I mentioned this, and so let me just specify exactly what has been done. There are two studies in which they have biopsied a few cells from the surface, and this is something which is done normally in the clinics. They check sometimes for diseases which are running in the families. And so they check the ploidy at the same time. And so they checked whether in the biopsies there were cells which are excluded or not. And then they see that when there's an excluded cells, there's more aneuploidy. But they sequence all the 3, 4, 5 cells that they are biopsying. So it is not formally shown that these are the excluded cells which are aneuploid. Okay? So we want to do this. We want to make sure that the cells that we measure the tension of are the aneuploid one. And then if you look in the literature about how cells become aneuploid, defects in contractility are also one of the causes of aneuploidy. Because if the cortex is too soft, and then the spindle cannot efficiently pull on itself, and then the chromosomes will have issues being segregated. So whether the cells are soft because aneuploid, or whether they are aneuploid because they are soft, is something that we would really want to find out.
Questions?
I have a more philosophical one.
What was philosophical?
No, because it's really cool that you worked on human embryos.
Yeah.
Like, it's not a model system, and I think recently there was a debate, and somehow someone said the chicken is the best model system. And so, from the behind the scenes of working with both, like, what is your opinion on having a mouse as a model system, and just the situation?
So from the behind the scene, I can even tell you that in this review, the chicken embryo was not there in the initial submission and was added after the revision. And somehow one of the reviewers was a prominent chicken researcher. But so then, I mean, I don't think everything should be done with human. I think, I mean, we, in the lab, we are very much interested in embryonic development in general, not in the one of the human. It just happens that many of the experiments that we do with the mouse, we just could technically do them easily in humans. So there was no reason not to do it. But I mean, as you can see, I'm interested in rabbit and cow development and evolutionary comparison. This is not something that we are really, keen on doing. But of course, if we can help people having babies, sure. Yeah.
Okay, so thank you for the interesting talk. And it's a follow-up question, actually. So how much do you find it encouraging or discouraging these ethical limitations?
The ethical what?
These ethical limitations, how much do you find it encouraging or helpful, or maybe the other way?
So I mean, in France, you can do quite a bit with human embryos, and what's limiting to some extent is how many embryos you can have access to. But legally, you can do quite a bit. I mean, in France, there was the law was changed this year. You can actually do CRISPR on human embryos for research. You can do quite a bit. And for what we want to do, which is studying the formation of the blastocyst, it's not a strong limitation. Was that your question?
Yeah.
Yeah, yeah. So in France, it's fine. There are other countries where you can do more, but don't need.
Thank you.
To which stage can you go?
So
in the development?
Yeah, so in France, the limit was pushed now to 14 days. 14, yeah. So in principle, that is after implantation. So technically, it's not even possible. Yeah. So it's not the law that is a problem.
Thanks a lot for this very interesting talk. So in this mechanical patterning idea, you've investigated the role of cadherin and contractility, but have you thought about the role of the protein that mediate the cortex to membrane attachment? Could they play a role also?
So we haven't looked at cortex membrane attachment. So there's little which is known about this. What we are planning to do is we are planning to measure this with optical tweezers and by pulling tubes of membrane at the surface. We are planning to do this, but we haven't looked at it now. There's not much known.
But don't you think that when you use the micropipettes, you're also somehow sensitive to the level of membrane to cortex attachment?
Yes. So what we have measured is that if you measure the tension of cells, so in the mouse, it's going to be about 400 piconewtons per micrometer at the end of compaction. If you block actin, for example, or myosin, you drop down to 50, 100. So most of the tension comes from actomyosin contractility. What's left from the membrane, I'm not sure.
But if you block ezrin or all kind of ERM protein family.
Yes. So this has not been done. We have not. No one has done this.
But maybe you would be pulling only membrane then, with your micropipette, if you block this coupling, mechanical coupling between the cortex and the membrane.
We could try with a pipette, but I think that would not be the best tool. I think pulling a membrane tube with an AFM or with an optical tweezer would be easier. We are going to do this actually next year. We're looking for people. Yeah.
Thank you. I have maybe a basic question, but at the end of the first part of the talk, you said that low contractility means both, I think. So low gamma CM and low gamma CC. So how to explain the extrusion of a part of one cell from the embryo if you have both low gamma CC and low gamma CM?
So you have low gamma CM and low gamma CC if you inhibit actomyosin contractility globally. Now, in the excluded cell, I suspect that there is low contractility and that this cell does not contract neither at its cell membrane interface nor at the cell-cell contact. But it's connected to a neighboring cell which does have a high gamma CM and which is able to contract. And I suspect that this cell, which is contacting the weak cell, is able to contract on the contact and make a large gamma CC. That's what we suspect. And the data are going in this direction, but the sample size is so small that we cannot statistically exclude that, we cannot make sure it's happening.
Thanks.